Hi, my name is María Sofi Viracacha Maya. I'm a med student and I'm on third semester at UPVs. Today's topic is going to be how SARS-CoV-2 platelets-derived microRNAs enhance NETs formation by extracellular vesicle transmission and TLR78 activation. Now, talking about their objective, it was to evaluate how SARS-CoV-2 platelets-derived microRNAs would enhance NETs formation by extracellular vesicle transmission and TLR78 activation. How did they do this? By exploring the roles of EVs in NETs formation from different types of patients. For example, they gave us this graphic to explain the situations. In mild asymptomatic patients, we're going to have a normal reaction in which there's going to be a production of EVs that are going to carry the microRNA and are going to cause the cell infection, and it's going to be like a normal reaction. But in the other hand, we have this situation in which there's a severe or moderate patient, and here we can see how there's a hyperactivation and a hyperproduction of EVs and they're going to activate the macrophages, and it's going to cause a cytokine storm which can provoke a thrombosis. So there are a few things I want you to remember through this whole presentation. Talking about SARS-CoV-2, this virus has a single-stranded RNA which is positive, and it will behave like a messenger RNA. It also has a spherical shape, has a lipid bilayer, and it has a diameter that goes from 60 to 160 nanometers. It does not require any DNA intermediaries to produce proteins for assembling new viral particles, and it also has a special mutation in its amino acid ASP614 to Gly614. So how does this work? It has a spike glycoprotein that it will attach to AC2. Normally in our bodies we have angiotensin II that promotes the release of noradrenaline. This will lead to vasoconstriction. AC2 takes this because it recognizes it and converts it to angiotensin 1,7 that will cause vasodilatation. So when there are mutations or when we block this AC2 receptor, vasoconstriction will occur and there's not going to be any vasodilatation. This is how we can explain many of the symptoms that are experienced during COVID-19, like difficulty breathing or shortness of breath, persistent chest pain or pressure, severe headaches of dizziness, persistent fever, severe cough. Also, this is how hypertension is likely to occur, and there may be poor regulation of blood pressure, and it can worsen cardiovascular function in COVID-19 patients. Talking about TLR78, these are toll-like receptors. They are integral membrane proteins, and they are located in the endosomal membrane. Here we have the ones that are of our interest. So they're going to recognize viral nucleic acid, like the ones from the SARS-CoV-2, because it's a single-stranded RNA. So when the cell infection happens, you know that the EVs are going to carry the microRNA that is going to cause the cell infection. It's going to also produce the production of NETs, but it's also going to activate these receptors because they're going to identify this RNA. And it's going to cause the production of pro-inflammatory cytokines. The methodology used in this investigation is PCR. It aims to amplify and detect specific DNA sequences in a sample. It is based on the ability of an enzyme called DNA polymerase to synthesize new complementary DNA strands to a specific DNA sequence. It is used in a wide range of applications, such as genetic research, diagnosis of infectious and genetic diseases, and identification of pathogenic organisms in clinical samples. Now, talking about flow cytometry, it analyzes and classifies individual cells based on different characteristics, such as size, complexity, and the presence of cellular markers. A flow cytometry is used when you want to get a quick and precise measurements of the physical and chemical properties of cells as they flow through a laser. So it studies the expression of proteins in the individual cells, and it analyzes the cell viability and conducts cell proliferation studies, and among others. Now, immunofluorescence assay. This test evaluates the antigen-antibody reaction to identify the presence of a protein in a sample. This protein is identified using an antibody labeled with a fluorochromosome. In the case of SARS-CoV-2, signaling pathways have been identified as mentioned earlier. During the research, the aim was to evaluate this pathway and its response by observing the reaction of TLR78. Immunoblotting, also known as Western blot, it is a test that detects specific proteins in a tissue sample. This technique is based on the separation of proteins by size through gel electrophoresis and their subsequent transfer to a membrane. Once on the membrane, the proteins are detected using specific antibodies to bind the proteins of interest. It can be used to identify viral proteins in samples from infected patients, which will help to confirm the presence of a virus and study its behavior under different conditions. In the results, we have a comparison between healthy subjects and infected patients. Various tests were conducted. For example, some toll-like receptors were blocked to observe which ones were more activated. For instance, it was observed that TLR7 was more pronounced than TLR8 or 9. Another example we can see is E1, an immunoglobulin analysis of COVID with health control. Here, CD81, CD63, and CD9 are markers that identify the presence of extracellular vesicles that lead to the formation of nets and activate the TLR78, which will trigger a cascade of virus response. So therefore, in the COVID culture, these markers appear very intense because during the disease there are much activated than in healthy patients, indicating a virus activity. These results confirm the article thesis, which is that plasma transfected with COVID showed a higher presence of nets. The discussion. We're going to talk about different authors and we're going to see if their opinions matches the investigation. The first author said, the release of nets was attributed to the capture and elimination of pathogens. Yes, it's one of the conclusions they got to. The second author says, we observed that the circulating nets, DNA, and EVs were both increased in the plasma of patients with COVID-19 and associated with disease severity. This is also one of the things I explained to you in the beginning of the video. We saw how there's different cases and reactions in a mild asymptomatic patient and a severe to moderate patient. So yes, it matches the investigation. And finally, we showed that the SARS-CoV-2 spike protein directly induced plateau activation and increased PEVs release in a dose-dependent manner. Moreover, we observed that the spike protein-induced nets formation was dramatically enhanced in the presence of a spike protein-primed PEVs, suggesting that PEVs play a crucial role in hyperactive nets formation. Also, one of the things I explained to you in the beginning of the video, so it does matches the investigation. For conclusions, we've got that EVs derived from COVID-19 patients induce nets formation via endocytosis uptake. The SARS-CoV-2 spike protein significantly enhanced this process in the presence of platelet-derived EVs through the TLR78 activation. Also, from this investigation, we can conclude that multidisciplinary approach, including biology, microbiology, PCR, immunoblotting, immunofluorescence, and flow cytometry, those techniques can help us deepening our understanding of molecular mechanisms and underlying the hyperinflammatory response seen in severe cases like COVID-19. Also, one of the things that caught my attention is that they said that the MIR21 TLR8 axis could be used as a potential predisposing factor or therapeutic target for the severe COVID-19. This means that it could worsen or improve the patient's condition during the illness, being a pharmacological target for the immune response to the virus. The MIR21 is a microRNA that is transported and then it activates the cascade. So this is very important because it could mean a huge improvement in medicine.